record and get my screen over. So we're going to finish going through the nervous system and then get started on the brain. Lab this week will be mostly focused on learning all about the brain, different brain components, what they do, where they're located. So that's kind of what next week's quiz is really all about, is the brain stuff. Let me pull up my chat box so I can have that.
Brains.
Yeah, no zombies, please. There we go. Now I have my chat box in case somebody has a question. All right, so we looked at the neuron itself the last time, kind of talked about how we have the dendrites that are where we typically are going to receive the incoming message that's trying to create enough of an electrical change to make that neuron depolarize and convince it to essentially pass its message on. Sometimes the messages that come into a dendrite can be an inhibitory message. Sometimes they can be excitatory messages. So we'll go into that a little bit later on, but if it's inhibitory, then it's essentially going to make it harder for that neuron to be convinced to depolarize. If it's an excitatory message, it's going to tip the polarity of the cell so that it makes it easier to depolarize. It's not usually one single message from one axon to the next dendrite in the chain that causes something to depolarize. It usually needs kind of a bigger stimulus, either that same axon sending that message repeatedly, or a whole lot of other axons tying into that neuron that are also trying to convince it to basically depolarize and pass it on. And so sometimes those influences are competing. We'll have inhibitory messages and we'll have excitatory messages. And then in that case, they kind of balance each other out and they don't really cause a net change in the polarity of the neuron. But we'll get into that a little bit later on with that. So let's move on to some different neuron types. Similar to how we classified our joints, we kind of have structural ways of describing neurons and different neuron types, and we have functional ways of describing them. So this first way that we're kind of looking at on this slide is the structural way. So we've got either unipolar, bipolar, or multipolar. And I'm just going to move to the picture and we'll kind of look at how those look. So in order on the top, we've got this unipolar. And so this is a really common setup for a lot of our sensory neurons, in particular for our sensations of touch in the periphery, whether we're feeling if something is painful, sharp, hot, cold, any of the things that we feel with our hands, with our skin. Essentially, we're going to have neurons like this that are set up just outside the spinal cord. And that's where that message kind of first gets received. And so it's kind of like it's offline. You can see how we've got the single process coming in from the dendrites at the left side are essentially like the sensory receptors would be in the skin, let's say. And it's going to send that stimulus up that pathway, and they refer to it all as an axon, essentially. They don't, with this unipolar setup, they don't technically say that there's a distinct neuron in between the dendrites and the axon. We've got the dendrites, which are essentially going to the receptor organelles, and then that message is carried on what they just term as an axon itself. And so it does make a stopover into that cell body, so that the cell body can decide whether it's enough of a stimulus that they want to send that message on and into the spinal cord and ultimately up to the brain. So that's, like I say, most commonly seen with our sensory neurons. The bipolar one in the middle, it's fairly similar in how it's organized to the unipolar. It's just that neuron is directly in line, as opposed to being kind of off to the side a little bit. So that's kind of the main structural difference. Essentially, we have they have equal kind of abilities. We have the dendrite end and the axon end, and they're both fairly important as far as how the message is being sent on. These are found mostly in our olfactory cells, the neurons that help us discriminate what we're smelling, as well as in the retina, in the eye. So the bipolar is the least common of these structural types. The unipolar is more common than the bipolar. The most common type that we have is the multipolar. And so the multipolar is going to be kind of how all our motor neurons are set up. that help us activate muscle action, as well as all the interneurons. Interneurons are going to be basically neurons that are between different parts of the brain that help different parts of the brain communicate with each other, as well as we have a lot of interneurons in the spinal cord. So a sensory message might come in, synapse with an interneuron, and then that interneuron might pass that message on to a motor neuron to cause an effect. And so the big difference with the multipolar versus those first two is that we're going to have just a whole lot more dendrites per axon. Essentially, we only have one axon really in all of these cell types. In the first two, we kind of have one primary dendrite that's feeding in. But with the multipolar, we have a much greater ability for a lot of other neurons to interact with that particular neuron that we're looking at. So it gives us more communication pathways, has the ability to be affected and influenced by more different types of messages coming from other parts of the brain or other related neurons.
So the dendrite is just that one branch, the one, I don't know what the correct terminology is, the one connecting port to the cell body is the dendrite, regardless of how many fingers it has extended?
Well, yeah, I mean, the dendrite essentially, it means that's the receiving end of the message for the neuron. That's where typically we're going to try to get the message to enter into that neuron and have an ability to cause an effect. The unipolar and the bipolar have we have kind of branches off that dendrite at either end, but they're all kind of receiving the same type of information in there. But it just has one primary communication pathway. The multipolar just has a lot more pathways into it, essentially. But when we think of just having one particular branch, usually we think of that more commonly with the axon. Pretty much all of our neurons have one primary axon that's going to send its message out. But depending on the structural type, you might have multiple dendrites coming into it, or you might just have one. I don't know if that helps. All right, let's look at the structure, or excuse me, the functional type. So we've already kind of alluded to some of these. We've got sensory, we've got motor, and then we've got interneurons. So the interneurons are neither going to be sensory or motor. They're basically just the go-between, kind of the middleman that's passing that message on. And most often it's going to be passing it on between the sensory message that we're receiving and then the motor output that we're trying to perform. And we have what we call sensory integration in the body. And if you think about any kind of movement, if you're walking on an uneven surface, let's say, if the ground I'm walking on is really, really sloped, one of my feet is going to be very inverted. The other one's going to be kind of more everted, essentially, because my legs are going to be upright. Otherwise, I'd be leaning over. And so I'm getting different type of information from my left foot versus my right foot. If I need to activate certain muscles on the medial or lateral side of my leg to try and help better control my ankle, I need to be able to interpret that sensory message that's coming in and create a motor response that's going to let me more strongly activate the lateral muscles on my leg on the foot that is kind of on the low side of the hill, and more activate the medial side of the muscles of the foot that's on the more high side of the hill. Because that's what's going to kind of help us control our balance and keep our ankles stable when we are on that uneven surface. And so those sorts of interactions take place all the time. And they happen. And when they don't happen, then people have a hard time moving because they're not recognizing what forces are acting on them in the outside world. We have a lot of problems with kids with autism in particular that have sensory integration issues, where especially textures and feels of things, loud noises, things like that, they have a hard time discriminating and tuning out what's not important and honing in on the important things. And so it does cause a lot of problems when those things don't mesh quite right. And the interneurons are kind of going to be that primary intermediary that's going to pass that on. We don't have a lot of cases where the sensory neuron is going to directly synapse with a motor neuron. There are a few spinal cord reflexes that work that way, but in most cases, you're going to have at least one interneuron in the chain that's just involved with passing the message on. So those are kind of our three different functional types. All right, so here's kind of an example of our motor neuron, and we're getting, I'm not sure why they're showing it on the person's skin in particular, but if you think about reflexes, you can do a reflex on biceps. Most people are familiar with the reflex on the patella tendon. That's the most common one that's checked, but you can do it really easily on the biceps too. And so if in this example, if we're looking at this diagram, if I strike the reflex hammer on my biceps tendon, it's going to cause a quick little jerk on my muscle, which activates a part of the sensory receptor in the muscle called the muscle spindle. And that muscle spindle is going to generate a protective mechanism. It feels that quick stretch, and it's worried that that might damage the muscle. And so the protective response is to cause that muscle to contract. And so that's why when you get that patellar tendon tapped with that reflex hammer, it makes your leg kick out. Your quadricep is contracting as a protective mechanism. And so this loop that's described here is basically kind of showing that reflex response. I'm not sure why they're showing the sensory part up here at the head or neck. But if I get that sensory message from the muscle that says, hey, there's a quick stretch going on, it's going to have that unipolar neuron in what's called the posterior root ganglion just outside of the spinal cord. So we see that kind of offline neuron with the inline axon / dendrite carrying the message all the way over. The purple interneuron inside the spinal cord is just receiving that message and then passing it on to the motor neuron that then tells the biceps to fire and activate it and cause that protective mechanism. So that particular diagram, we don't have to think about it at all. It doesn't have to go up to our brain for our brain to say, hey, why don't you contract biceps? You might be in danger. We need it to happen faster when we're looking at these reflex responses. So we will get the message to the brain, but it's a little bit slower. And so if we are in a really crisis, dangerous situation, we want that response to be even faster. Essentially, it bypasses the brain and just the spinal cord takes over with that. So that's kind of an example of how we might see those things. Again, the interneuron is a multipolar neuron. The motor neuron is going to be multipolar. We don't see that bipolar. Again, the bipolar ones are pretty much just in the olfactory neurons as well as in the retina neurons. So those aren't really common. All right. So we talked about our neurons. That's our primary cell in the nervous system. The other types of cells that are in the neuron systems are going to be the helper cells that in some way support the function of the neuron itself. And so we call these glial cells or neuroglia. And we have different types for the central nervous system, the brain and spinal cord, versus the types that we have in the peripheral nervous system, kind of more out in our body. And we do have tons of them. Neurons, we talked about last time, are generally non-mitotic, so they don't have cell division taking place. Most of our cancers are a result of a defect in cell division, where we make a bad copy of a cell and the body's response is, oh, that one's not right, let's make another one. And if the blueprint's wrong, which is what happens with most cancers, the DNA replication process is flawed, so we keep making flawed copies. When people get brain tumors, they're pretty much just going to be in these glial cells because the neurons themselves don't replicate. They don't have the ability to have defects in that regard. If you have defective neurons, generally you're born with them or maybe you damage them, but when you damage them, they don't really heal themselves. But these glial cells, they can get damaged. They have a normal lifespan, so it is normal for them to divide and make new ones to replace the old ones or to replace ones that are damaged. But when that process goes haywire, that's when we start getting brain tumors or spinal cord tumors.
So that's where glioblastoma comes from.
Yes. Yep. Glioblastoma, astroglioblastoma, I think is what John McCain had, and so that affects the astrocytes. And so sometimes they will name it based on which particular glial cell is having that problem, but glioblastoma could mean any of these four types might be creating a defective copy and making that tumor. Yep. So here's our 4 types that we're going to see in the CNS. So supporting the neurons in our brain as well as in our spinal cord. We've got astrocytes, ependymal cells, microglia, and oligodendrocytes. So I'm just going to go through, I'm going to look at this one picture here, and we'll just kind of talk about what they all do, and then I'll skip ahead a few slides. So this diagram is showing all 4 glial cell types. It's showing the neurons themselves. You can see the 2 kind of ones that look like a pair of eyes kind of right in the upper portion of this picture. The green ones over here are the astrocytes. So they're somewhat star-shaped, and that's where the name comes from. I don't think they're actually green. They just show them that way to make them more distinct in the image. Their primary job is to establish the blood-brain barrier, if you've heard that term before. We don't want harmful chemicals to get up into our brain because those neurons don't replicate. If they get damaged because we just let anything pass through the blood and get to our brain, we've got problems. And so these astrocytes, you can see all these little things gripping onto this capillary, and they call those perivascular feet. And essentially they're surrounding the perimeter of that capillary. And they're basically creating only small gaps in that capillary wall. Things like oxygen and carbon dioxide, those small gases, are going to be able to diffuse out between those spaces, but bigger structural molecules, chemicals, hormones, proteins, things like that are going to have a much harder time getting past those. It's basically like a fence. It's restricting what can pass through that capillary. And that's essentially how it works. And it's both good and bad. When people need a medication to get to the brain, because say somebody's got epilepsy or something like that, and they need to get the medications to calm that response down, they have to design the medication in such a way that it has the ability to get through there. Otherwise, it's not going to be able to pass through there. So it's a good thing that we have it. And if astrocytes aren't working right, then we're going to have problems because things are going to get into the brain that we don't want to get into there. So that's the astrocytes' job. These little blue ones up at the top, got a few other ones spaced around that are just kind of hanging out independently. Those are our microglial cells. These are immune system cells that are basically kind of the first line of defense if something harmful does make it through that capillary. Then the microglial cells, if we get a virus or a bacteria that potentially got through our blood, those would be the things that would try to immobilize it, try to activate some backup, call in the cavalry, the other parts of the immune system that would kind of help it combat whatever was invading. But they're kind of the sentries that are kind of in that neural space, again, in between the neurons, but hanging out and basically being the guards. They're the security guards in case something else comes through that's could potentially harm things. In the center with these multiple legs, they kind of look like blue neurons a little bit, but then they have all of these projections. And those little projections are creating the little myelin wrappings that go around the axons for all these neurons. So our oligodendrocytes their primary thing that they're doing is creating the myelin for the axons so that the neurons that are trying to send messages out to other portions of the body can get there faster. Again, that myelin is going to help speed that rate of conduction of the depolarization signal. And we have a similar structure in the peripheral nervous system, but it's a little bit different. These oligodendrocytes, you can see the one that I'm kind of moving my cursor over, it's got 4 different sections of myelin on 2 separate axons that it's connecting. In the PNS, each individual section of myelin is its own cell. In the CNS, each little individual section of myelin is a part of an oligodendrocyte that has multiple myelin wrappings as a part of itself. So it does kind of bind these axons kind of together. Probably helps to organize them a little bit. But its main purpose is to try and insulate the wiring so that electrical signal can be sent out a lot faster. And then the last of the PNS glial cells are these ones on the perimeter here, these ependymal cells. And these are secretory cells. So these are what are going to make our cerebrospinal fluid. The cerebrospinal fluid is made from plasma. It doesn't really have blood cells in it per se, but it's got a lot of ions. It's got basically like a lot of water. It serves to make the brain and the spinal cord kind of buoyant inside the skull, so it kind of floats it in there to protect it a little bit, give it a little bit of cushioning. But it also helps with delivering some nutrients, picking up some waste products. But that CSF is a crucial part of keeping the brain essentially well lubricated as part of its function, and also to serve as a protective barrier essentially from forces. So those are our 4 glial cells that we've got in the CNS. The astrocytes for the blood-brain barrier, microglial cells are our immune system cells, the oligodendrocytes creating the myelin to help speed conduction, and then the ependymal cells that are producing cerebrospinal fluid. So those are our 4 types. All right, let me scoot ahead here. All right, PNS, we've only got 2. So it's a little simpler. We've got what are called satellite cells, and these functionally are kind of like the astrocyte. Their job is to kind of help maintain a healthy surrounding environment chemically for the neurons to be viable and thrive and have the support that they need. The neurolemmacytes are what used to be called Schwann cells, if you've heard that term. Those are essentially the myelin wrappings that surround the peripheral nerves. So 2 types, easier on the PNS to keep straight. So here we're looking at our posterior root ganglion. Again, that's where the sensory messages from the periphery of the body are going to come into the spinal cord. And we've got those cell bodies, those unipolar cell bodies that are located there. Surrounding each of those unipolar neurons is going to be a cluster of these satellite cells. And again, their main job is to try and just maintain a nice homeostatic, chemically balanced environment for the neuron itself to keep it healthy. And then our other type we see are these little wrappings. Those are our myelin sheaths. And we'll look at that real quickly. So our neurolemmacytes, whoops, I skipped ahead too far. Right there. We don't have a central neuron that has multiple attachments like we saw with the oligodendrocytes. Each individual section of myelin has a neuron, has a nucleus. It is a cell. So it's an individual cell, whereas in the CNS, they're just a component of another cell. You can see that neuron is kind of on the perimeter, and it does just wrap around it. So if you imagine like a tube of toothpaste that's getting low, if you squeeze it from the end and then start rolling it upon itself, that's essentially kind of how these work. They coil themselves around that axon to create multiple layers of insulation, and then all the fluid, all the cytoplasm and the nucleus all kind of get compressed and concentrated into the perimeter. We can't compress the nucleus, otherwise it would die. So we want that nucleus to kind of still be viable. So it still has a little bit of fluid in there to help it kind of keep its functions going. And if it has to send things out of the nucleus to make a product or whatever, it can do that. But these are functionally the same as an oligodendrocyte. They're just organized a little bit differently. They are each an individual cell. All right, so again, I've kind of mentioned already that myelin, it's basically fat is kind of one of the primary components of it. And fat's a really good insulator. So it works really well to help us speed up the rate of electrical transmission so that our nervous system is quicker and more responsive. When you're a kid, a lot of our axons are not fully myelinated yet. And it has a large effect on our ability to be coordinated with certain movements. If you think about a little kid learning how to throw a ball or something like that, the first few times they try to do it, they're not very good at it. If they're trying to catch a ball, field a ball, things that require a lot more dexterity and a quick response with accuracy, they're going to struggle with, because many of their neurons are not myelinated. Certainly they haven't practiced that movement yet to gain proficiency, but they're also limited by how fully myelinated those neurons are. So sometimes kids that have maybe a little quicker development along those lines can be more skilled and coordinated at an earlier age. And it does give them an advantage. And certainly when you look at kids that are competing, you see kids that are all like, let's say a 7 and 8 year old soccer team or something like that. Some of those kids are way more coordinated and developed than others. And a lot of it has to do with how well myelinated their neurological system is at that point, especially in those younger ages. All right. These are just some pictures of how we myelinate and kind of wrap things up.
Is there anything that can be done nutritionally or therapeutically to accelerate that process?
I don't think so. I mean, unless they've identified like a certain enzyme deficiency that the kid has that might be limiting their ability to produce myelin. I mean, I guess if people really were very much in malnutrition, they might have issues. But I think if you're getting a pretty normal diet, you probably have plenty of fat to help create those myelin cells, whichever type you're looking at. But there are probably some, like say, if you're missing a particular digestive enzyme and you can't absorb something, or you can't create the cell wall in a proper way, it might cause a problem. But there's not any that I'm aware of. On this particular slide over here, we've got this term saltatory conduction. And so if we look at the bottom picture, we've got the dendrites all around the perimeter of that cell body. Let's say I'm sending a lot of excitatory messages through a lot of other neurons that are telling this neuron, hey, this is really important message. You need to depolarize and fire it and send it along. Essentially, the resting membrane potential of these neurons is, usually they estimated it to be like -70 millivolts. When we get enough of a excitatory message to cause that negative value to climb up by about 15 points, so when it gets to about -55 millivolts, that's considered to be the threshold value. And when that threshold value is hit, it causes a lot of what are called voltage gated channels to open up. So we talked about channels in a cell membrane, how they can have gates that will open or close to let certain things in or out of the cell. A voltage gated channel, essentially when the electrical current level reaches a certain value, that's going to trigger it to open. It's going to enable that circuit and it's going to open that pathway. And in this case, when that -55 value is reached, a whole bunch of sodium channels open up, a whole bunch of sodium rushes into the cell, and that causes that polarity to go from -55 at that threshold to +30. And when it hits that +30, that's going to create what's called an action potential. And that action potential is essentially a wave of positive electrical activity that is now going to be sent out that neuron's axon and travel down to the end to send the message on to whatever is at the end of that axon, whether it's another neuron, whether it's a muscle, whether it's a gland, whatever the case might be. So when we get that message going down a myelinated axon, instead of the electrical wave of polarization having to travel along the whole way, it basically gets to skip. And the axon only depolarizes where it's not insulated. So only in the little gaps between those individual neurolemmacytes is where the axon is actually having to create an electrical change and open up gates and close gates and things like that. And so that process of opening and closing gates takes a little bit of time. And so if we can speed it up, essentially be like a speed reader, and we know where that message is going, there's no need to kind of open all the gates in between. It'll go faster if we just let it skip ahead. So that's kind of what we call saltatory conduction, when it's able to just hop and only depolarize the sections of the axon that are exposed. If they're covered with the myelin, they don't need to change the polarity in that section. When we have an unmyelinated axon, it has to travel the whole way through there. So it doesn't get the ability to skip ahead. Think about like I was just kind of thinking about, think you're at an amusement park, and when you get there first thing in the morning, you've got those little turnstiles where you have to wait in line. You go right and left, kind of weave your way up to the front of the line for that great roller coaster. If it's not busy, they might have the first like 5 rows of that back and forth kind of blocked off. So you can just go straight to the front of the line. That's essentially what myelin does for us. It lets us get that message straight to the front. If we don't have a myelinated axon, then it's essentially that message has to go the long way. Even though we know that there's nothing in the way, there's nobody in front of us in line, we still have to go that whole way, all the way back and forth. So it's just a slower process, and slow is relative. It still is happening really quickly on an unmyelinated neuron. It's just way faster if the neuron is myelinated. So it gives us a better ability to respond quickly and react more quickly and receive that information more quickly too. So anytime we have something that's dangerous and potentially harmful for us, we want that message to get to us as quickly as possible. So myelin's our friend with that. So here's our unmyelinated one. These are still have some myelin around them, but because they're not completely covered, we have an exposed surface of the axon, and so that whole thing has to depolarize bit by bit. All right, now axons in the periphery are pretty cool. We talked about how neurons cannot regenerate themselves, and that's true for the most part of all the cell bodies. We can't make a new nucleus, we can't do cell division, all that kind of stuff. But if a peripheral nerve is damaged, and this only works in the PNS, it doesn't work in the CNS, but in the PNS, when a peripheral nerve is damaged, it does have the ability to regenerate its axon so that its message from here or from the spinal cord can still be carried on. And I had this happen to me once. I've had it happen to a friend of mine too that put his arm through a window. But it does depend on where the damage is, how bad it is, and how much destruction of the nerve itself there was. It is a slow process. I'm not sure if this last bullet point is on the slides that are up on Canvas. I may have added it after the fact, after I posted those a while back. But the rule of thumb that I remembered is it's roughly about 1 inch a month that it takes for an axon to regrow. And so a number of years ago, before I was a PT, I had been having some problems with my left knee, and I had an ice pack on the left side of my knee on the outside, and I put it on. I was holding it in place with an ACE wrap so it wouldn't slide off. Right over my fibular head, we have a very superficial nerve called the fibular nerve or the perineal nerve that's right there. Think like the funny bone kind of for your elbow. It doesn't take a lot. There's not much padding over the top of it. And so I basically froze that nerve right there. And for literally about a year and a half, the lateral side of my foot was numb. It had like that pins and needles sensation to it. And it took literally about a year and a half because it's about a foot and a half away from where I damaged the nerve. And everything below that, it was not able to send that message or receive that message accurately. I guess it was more just the sensory part that got damaged. I didn't really lose function. I could still evert my foot. So I didn't damage the ability to activate the muscles, but the sensory nerves in particular were affected, and it took a really long time to come back. So we do have to be cautious about things like ice and heat, especially in areas where we have, or compression in areas where we have superficial nerves. Definitely a problem. So here's kind of what it looks like. If we get damage on that axon, as long as what's called the neurolemma, that kind of outer sheath, stays more or less intact, then everything distal to the site of injury degrades, and then we're able to reform those, and the axon is able to basically grow back out through that channel. But if that channel gets disrupted in a significant way, it's not going to be able to do that. I had a friend in high school that put his arm through a window and he cut his ulnar nerve right here. And they went in and did surgery right away and reconnected those nerve ends so that this pathway would be there. Had they not reconnected those nerve ends, there would have been no way for those axons to find their way to where they're supposed to go. So they really, in that case, because it was such a big, thick, major nerve that's carrying a lot of different axons, they had to surgically reattach it. And I'm sure it wasn't aligned perfectly, but the body still was able to figure out which particular axons needed to connect where distally to go where it needed to go. But that guy's hand, that whole side of his hand where the ulnar nerve distribution is, was white and scaly, atrophied. He lost muscle function. Again, because even though they reconnected stuff, when he cut it, everything distal degraded and then had to reestablish and regrow back out to that distal part of his hand. So it was a long time coming for him to get function back.
Hey, Craig, I have a question on that.
Uh-huh.
When you have that nerve reconstruction, does that increase or decrease the sensitivity of it?
Yeah, you're likely to have some lingering deficits from something like that, either on the motor side or the sensory side, or potentially both, where you might not have.
Does that mainly like depend on the person?
Yeah, it would depend on the person. It would depend on how severe it was, and it would depend on how well able to really line things up precisely on the two ends they were. You know, if they were lined up at 90-degree angle to each other, it would probably have a harder time than if they'd lined things up more directly in line with how it used to be before it got cut. And so when you have something like a nice clean slice, like a piece of glass or a knife, it's easier to do. If you have something like a car wreck where you have a lot more shearing and trauma and compression and bruising, it's probably gonna be a lot harder to make that nice clean reconnection. So you're probably more likely to have some of those lingering problems when it's kind of a more gnarly, chewed up, kind of less surgical type of injury with that. All right, so when we talk about a nerve is really a grouping of axons. It's not just one specific axon, it's a whole collection of them, and we kind of bind them together to make them more well organized in the body, so that similar information going and coming from similar places is kind of all in this protected pathway. Think about, you know, underground internet cables or phone lines or anything like that. You got one primary branch coming into the neighborhood, and then you're going to have a whole lot of little feeder lines kind of splitting off of it, so that that service can be carried to all the homes. That's kind of how our nerves are set up. We've got that central passageway that's got a whole bunch of stuff there, and then when that individual axon gets where it's going, it's going to hit the off ramp and it's going to split off and go where it needs to go to either be able to send the sensory information from the periphery out or to the brain, or send a motor movement message out from the brain. All right, so very similar to how muscle is structured when we look at how these nerves are set up. We've got an endoneurium that's going around an individual axon. In the muscle, we have the endomysium that was going around the individual muscle fiber. We've got the perineurium that's going around a group of axons that we call a fascicle. A group of muscle fibers is also a fascicle that has the perimysium going around it. And then we have multiple fascicles together, then we're going to have another surrounding outer layer, and that's going to be the epineurium. Just like in muscles, we're going to have the epimysium. So same exact terminology, we're just changing out the mysium for the neurium. But the organization is very, very similar. And that's kind of what we're looking at here. We've got a primary blood vessel, arterial side and venous side that's helping supply that nerve with nutrients along its way. And then we've got, again, individual axons surrounded by the endoneurium, fascicles that have the perineurium, and then groupings of fascicles that are going to have the epineurium surrounding them. So that's kind of, again, how our muscles are very much organized also. All right, let's talk about our synapse. And this should mostly be really a review. I'm just going to go ahead and go to the picture. This is very much like we talked about with the neuromuscular junction. The only difference really is that instead of this being a junction between the end of a nerve and the start of a muscle, synapse is just from one neuron to the next neuron. So essentially where one axon is going to be touching down on another neuron's dendrites to cause some effect or be able to pass that message on. And the diagram below is kind of the shorthand way that they often draw this to kind of show how these messages are being passed on. The upper illustration is supposed to be more the anatomically kind of correct one. And these synapses can be at lots of different places. Sometimes the axon will synapse directly on the neuron cell body itself. Sometimes it will synapse on the dendrite, and sometimes it'll synapse on another neuron that's coming in on its dendrite or on its axon to try and cause an effect. So we do have a few different places where these synapses can take place, but ultimately it's just trying to pass that electrochemical message on from one axon to the next neuron's dendrite or body to try and stimulate it to either be inhibited or to be excited, depending on if it's passing along an excitatory message or an inhibitory message. It's easier to think about the excitatory messages. That's what's going to cause a depolarization. An inhibitory message makes it harder for that neuron to depolarize. All right, so this is showing kind of some different ways that these synapses can take place, either on the cell body or most commonly on the dendrites of the receiving cell, but then sometimes it will be kind of more axon on axon, which is not as common. All right, so this should look fairly similar, at least the one on the right. This looks very much like our neuromuscular junction. We've got the axon coming down. We've got the synaptic bulb, this kind of bulbous ending to it. Inside of that synaptic bulb, we have vesicles that cell has produced that have neurotransmitter in them. The neurotransmitter is just a chemical that is going to cause some sort of reaction on the cell that it touches down on. When we have a wave of depolarization coming down that axon, that change in polarity will open up these calcium gates that you see on the left side of this image. That change in electrical activity opens that up, so now calcium can come into the end of that synaptic bulb. That calcium is what causes these vesicles to recognize, oh, that's our signal that we're supposed to go. Think about like paratroopers on a plane, and all of a sudden that lead guy is like, okay, go, everybody go, 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 go. That's calcium's job. Calcium comes in and is like, okay, everybody go, 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 go. And so all these vesicles start moving towards the end of that axon so that they can merge with that cell wall, release their contents, which is that neurotransmitter. Neurotransmitter is going to flow across that gap, that synaptic cleft. So it's not directly in physical contact. There's a small space, flows across there, moves from high concentration to low concentration, so it diffuses, and it will bind to receptor sites on the receiving side of the cell, on the dendrite or the soma, or whichever part of the cell this is touching down on. When it binds on that receptor site, that's going to open up some gates on that cell wall, which let positive ions, usually sodium, come in to cause that cell to depolarize. So sodium is really the key thing that causes depolarization of the cell as a whole. The calcium gates are really important in helping mobilize the neurotransmitter release. But sodium is the key ion that is going to cause these neurons to depolarize. We open up gates that let a whole bunch of positive sodium ions in, and so the inside of the cell goes from being negative to being positive, and that's what we mean when we say depolarized. It switched its polarity from being negative inside the cell to now all of a sudden it's positive inside the cell because we let in a ton of sodium ions. And it's brief, and we basically depolarize that particular section of the cell, and then because we've let all this positive ion in, it triggers a bunch of voltage gated channels adjacent to that synapse site to open up. And when those adjacent ones open up, now a bunch of sodium rushes into that section. So if we imagine like along my pen, let's say that I have that action taking place right here. I'm getting a section of that cell wall that's depolarizing right here. That's going to trigger sodium gates here to open up. When these sodium gates open up, now sodium rushes into this part of the cell. And when that happens, it depolarizes here. And that's going to open up sodium gates right here. And so it just keeps going all the way down. And that's what lets us depolarize and send that message along. And so when we talked about myelin earlier, if I have sections of myelin, now instead of having to open up gates along that whole length of that axon, I can just essentially go, yep, 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 quickly send that message on a lot faster. But it isn't causing the entire neuron to depolarize right away. We call it a wave of depolarization. And so just like a wave in the stadium where one section stands up and raises their hands and then they sit down, we're going to depolarize one section of that axon. It's going to pass its message on, and then that first part that depolarized is going to reset itself. So the next time that message comes down, it's going to be able to be depolarized again. So it is a brief period of time that the inside of that section of that axon depolarizes, or that cell body, whichever part is in contact. But that's ultimately how that depolarization process kind of takes place. Over on the left side here, we have the more atypical type of synapse. This is a electrical synapse. The most common ones are just chemical. So these chemical ones are triggered by neurotransmitters. The electrical ones, we're just letting that charge pass on through, essentially. And these are pretty rare. One of the more common places that we'll talk about it is in heart muscle. We talked about heart muscle a couple weeks ago briefly. We had the intercalated discs that were kind of the little sections. These were, the intercalated disc is the electrical synapse. So that when we're trying to make the heart contract in a nice synchronous fashion, these pathways let that message be traveled more quickly than if we were having to depolarize the entire muscle. That would be a slower process. So it's basically like a dedicated electrical passageway for this depolarization current to move through from one cell to the next. So again, it's just made to speed things up and help the heart contract in a more synchronous and efficient fashion so we can pump blood with less work and better organization. So those are our synapses. Again, chemical is the far more common one, and that is basically the same process that we see at neuromuscular junctions. The electrical one is much less common that we're going to see. All right, we're just about done with this chapter. Let me just go through these real quick, and then we'll move into the brain so we can at least get through part of that before we get to lab. So these circuits that we see here are really just going to be found in the central nervous system. And we've got different reasons why we might have neurons organized in these different ways. This converging circuit is essentially, we have a whole bunch of individual neurons. All these blue neurons are trying to make this red neuron fire. And so this could be like a, let's say we see a fire, okay? Everybody had fireworks. We've had a number of wildfires going around. As soon as you walk outside, if there's a fire going on, you might see that the air is kind of hazy. And so maybe this one over here is the vision one that's saying, hey, it's kind of hazy and foggy out here, like there's smoke in the air. This one is for our nose, let's say, and we're like, oh, I smell flame. This one is for our ears, and it's like, oh, I hear this crackling, burning wood sound. And this one is, I don't know, something else, the wind. Maybe we're feeling the wind on our skin, and we're going, oh yeah, it's coming from over there. I can feel the wind on my head. So all of these things are sending us information about that fire. And then this neuron is like, I'm going to conclude that there's a fire going on. And so I'm going to send this message to my legs and say, let's book it and get out of here because I don't want to burn. Or let's hop in the car and get out of here. So this is all different information, but it's all trying to get the same response. It's like, hey, dude, there's a fire. You better get out of here. If we've got a diverging circuit, this is a good way for our muscles to work. Let's say there is that fire. If this is, let's say this neuron here, let's replace it, and now it's this blue one up at the top of the second image in this diverging circuit. Let's say I need to activate my quadriceps or my hamstrings. Big muscle groups are going to help me run away from that fire. I don't want to have to send an individual message to each particular motor unit. I want that message. I want to send out one message and have it just go out to everybody. Think like an email blast. I don't want to have to write an email to every single one of you. I'd rather just put it into Canvas and send it out to everybody at once. It's more efficient that way. So when we want to send that message far and wide very quickly throughout the body, a diverging circuit is a good way to have that. We have one central message. It's going to be passed on to 3 friends, and then they're each going to pass it on to 2 or 3 friends. It's kind of like the old phone chain for, Tom and Allie might remember that. The rest of you are probably too young. But basically, we would just have these calling trees essentially to try and let everybody know that soccer practice was canceled or whatever. Maybe that's even too old for us, because even I had email for stuff like that for my kids. This was back when I was a kid, maybe when you were kids, that your parents had to deal with. So that's kind of a diverging. This reverberating circuit is often used for trying to help kind of maintain homeostasis a little bit. And the most common example, and the one I think the book talks about if you read through it, is with respiration. If you think about our breathing, when we blow all our air out, we have sensory receptors that basically in the lung tissue itself that say, there's not very much stretch on me. I'm pretty empty. It's probably time to stretch me. And so that basically sends a message that says, hey, the lungs are empty. We need to get some air in there. And so we start breathing in. And as we breathe in, we get to the point where the lungs are like, okay, I'm definitely stretched now. You can quit breathing. I'm not doing that much work. There's no reason to breathe really more deeply than that. And that's going to trigger it to basically shut off. And now we're going to let those muscles relax. So when we have a repetitive cycle in the body, like breathing or like the heart rate, these reverberating circuits are a good way for the body to kind of control it. They can be modified. If I start exercising, I'm going to need to speed those things up. I'm going to need to change how quickly I have inhalation and how quickly I do exhalation, how deeply I'm going to do those things, so that we can modify them. But the same pattern, basically, we're going to inhale, and we're going to exhale. We're going to inhale, and we're going to exhale. And so, but we need that extra nervous system information to guide those responses and figure out how frequently we need that motor response to take place. This parallel after discharge circuit is more kind of executive planning kind of thought process. If let's say we're reading a book or reading an article or doing something like that, we might see things on the page. We might think about how that relates to our past experiences or what we've read about previously. We're basically calling in other information that's helping us come to the same conclusion. So it is giving us, associations from different memories and different things, images, sensations, whatever the case might be. So it's similar to converging, but essentially we could get the same message individually multiple ways. So it's not as distinctly different of a message as we might see with a converging circuit. It's just kind of different ways to kind of think about it or pass it on. So those are kind of our 4 primary ways that we're going to be organized to kind of send these messages out. All right, I think that's it on this chapter that I really wanted to cover. We kind of already talked about the nervous system development earlier in the semester. So what questions you guys have on neurons? Anything on action potentials or depolarization that's confusing? We'll get into the chemical mechanism, and you have to know more about the specific values as far as the depolarization values in physiology. But since this is anatomy, the focus is more just kind of on understanding how these gates work, the fact that there are gates that help control the charges coming in. We want to understand how the neurotransmitter gets released and what effects those have. Different neurotransmitters will have different effects. The one in the image that we looked at was acetylcholine, and that's probably the most common neurotransmitter in the body, but there are a lot of other different ones too that we see. But acetylcholine is what activates skeletal muscle, so it is a big one for motor, for movement. But there are other types too. All right, so far so good? All right, let's look at the brain. We won't probably get to the spinal cord or anything like that. And we're going to talk about these 4 different regions, and then hopefully we'll at least get through the cerebrum. and that'll get you at least started on when you get into lab. It'll make a little bit more sense. It'll be the first time you're seeing these things. So the cerebrum, we get the diencephalon, we got the brainstem, and then we have the cerebellum. So 4 kind of primary big distinct areas. And we have 2 halves for the cerebrum. We're going to have 2 halves for the diencephalon. We only have 1 brainstem. And then we have 2 halves for the cerebellum, the right and the left, for all those other structures. When we look at the outside of the brain, I'm going to use my other model. That model kind of falls apart a little bit too easy. This one does too, but all right. So our brains are not multicolored like this. Sorry, that make it too easy to identify these structures. But this would be essentially the left half of the brain right here. It'd be organized like this. This would be the front. So on the perimeter, you've got lots of little grooves and bumps and things like that. So the projections that stick out are gyri, or a gyrus would be the singular term. A gyri would be multiple. The indentations in between are the sulcuses or sulci. And so those are basically, that's the soft tissue of the brain. Essentially, it's going to be the gyri, the sulci are those little indentations. And that's what we use to kind of help identify different parts of the brain. And supposedly it's kind of like a fingerprint. Everybody's is a little bit different as far as where their particular gyri are, but we all are basically put together the same way. Anterior is going to be this end. And posterior is going to be back here. And most of these lobes in the brain, these different colors that you're seeing on this particular model, are named based off of the different plates in the skull. So the terminology should be pretty familiar to you from when we learned the skeleton. All right, so I'm gonna have you guys look at this picture here. Again, this is the same thing, left side of the lateral view that you're seeing. The first part that we'll talk about is the frontal lobe. And so the frontal lobe is this whole big section. It's probably the single biggest part of the cerebrum. Well, let's back it up. We'll look at cerebrum right here. Cerebellum is at the back. This part that's got more of these kind of lines to it. It's not as much of a squiggle. They're more kind of distinct point-to-point kind of lines. The pons and the medulla are these larger kind of swellings that are coming down, and they're basically at the beginning of the spinal cord. Once we pass the medulla, then oblongata, then we're essentially into the spinal cord. And so the foramen magnum, that big hole in the bottom of our skull, is essentially right below that medulla and coming out right through there. We don't see the diencephalon in this picture. We can only really see it. It's very centrally located. When we look at the inner side, then we'll talk about the diencephalon. But when we're looking at the outside, we can't really see that particular structure. So all of the part that has the sulci and the gyri is the cerebrum. So this is the left cerebral hemisphere of this image of the brain. We've got 5 lobes. We've got the frontal lobe that's going from this central sulcus anteriorly. We've got the parietal lobe, just like we had the parietal part of the skull on the side. And that's going to be kind of a little bit more posterior, behind the central sulcus. At the very back, we've got the occipital lobe, just like we have the occiput for the bone. And then lower on the sides, below the parietal, we've got temporal. So that one makes sense too. So all of those, you've heard those terms. And they're basically just right where those 4 bones overlay the brain itself. In this lateral sulcus, this kind of deep section here, is where we'll find the 5th lobe, the insula. So the insula doesn't have a bone in the skull named after it. It's a deeper lobe kind of inside. There's one particular view, the coronal views of the brain, when we're cut this way, that's the best way to see the insula. But normally you really can't see it from the outside unless you retract it. If you pull the temporal lobe down away from the frontal lobe and the parietal lobe, then you'd be basically looking in to the neurons that make up the insula.
Are those lobes not connected to each other? They're just nestled inside the skull?
They are connected internally. So we do have passageways for axons to communicate from one lobe to the next so that we can kind of make sense of things and form memories. And those different lobes do different things for us. And so when we talked about that fire situation with those converging neurons, the neurons that would be sending us information about what we're hearing and what we're smelling are in the temporal lobe. The ones that are what we're seeing would be coming from the occipital lobe. The ones for like sensation, if we felt wind on us, would be coming from the parietal lobe. So we do have to have those interconnections between those different lobes to kind of make sense of things with that. But there is a big groove. I mean, they are, you know, you can put your finger, when we have the specimens, you could definitely put your finger down in between those sections and pull them apart a little bit. Now we have what's called the longitudinal fissure right along the top that separates the right and left cerebrum. That's really deep. I mean, you could stick like all your fingers down into that before you hit the barrier of the corpus callosum. And then where that lateral sulcus is on the side between the temporal lobe and the frontal and parietal lobe, that one is pretty easy to pull apart. The other ones are not as deep. They're definitely not as easy to find. All right, so this is looking at the underside of the brain. Essentially kind of looking at it this way. And so at the bottom of the picture, we've got the cerebellum. You're looking at the brain stem coming up. So we've got our medulla oblongata. We've got our pons. So the pons is bigger than the medulla oblongata. And then from the pons, we go up into what's called the midbrain. Now this image is not an easy one to see the midbrain, but essentially where these mammillary bodies are, these two little bumps, that's kind of where the midbrain is. All the things in yellow are the cranial nerves. And we'll go over the cranial nerves in lab this week. I don't know that we spend a ton of time in lecture. I think we'll get to them on Thursday for sure in lecture. But you can see the eyeballs. So that's the optic nerve going to the eyeballs. These two that are sticking out here that look like kind of little antenna, those are the olfactory nerves. So those are going to be coming anteriorly. And then when we get into our sinus passages, essentially it's going to drop nerve endings down through that cribriform plate. If you remember from our skull, here's my skull. So I'm looking at the anterior side right here. Right in here, we have the crista galli. It's kind of this distinct ridge at the front of the ethmoid bone, and it has all these little perforations in it. And those little holes, those little perforations are where the nerve endings for the olfactory nerve would come down and essentially drop down in to our sinus passageways to help us be able to make sense of what we're smelling. So that's the olfactory. We'll kind of go through the rest of them when we get to the cranial nerves, but that's what all of these are meant to be. Other things of note on the medulla oblongata, we have what are called the pyramids, and that's these two columns. I'm not sure who named them. I would have named them the columns because that's what they look like, but they named them the pyramids. And so the pyramids are really important. They are the most common place where axons cross over. So most of you probably know that the left side of our body is controlled by neurons on the right side of our brain and vice versa. Where that axon crosses, the most common place is in the pyramids, right there. That doesn't happen up here usually. Some cases, there are some that cross over in the spinal cord and then send the message up to the appropriate side of the brain. But most of the functions in the body are going to cross over in that pyramid. All right, you can see the temporal lobe from the underside through here. Again, you can't really see the insula yet until we get to the interior view. All right, here's this view. So essentially we've cut the brain in half. We're looking at the middle inside. And this is where we can see the diencephalon. So we've still got our lobes of the cerebrum up top. And then you see this kind of white section or kind of beige section here in the middle that I'm outlining. That is called the corpus callosum. And that's essentially a place for a bunch of myelinated axons to cross over from the left side to the right side of the brain. This isn't going from the right side of the body to the left side of the brain. it's just going within the brain. So it's helping the right side of the brain understand what the left side of the brain is interpreting and vice versa, so that we can get a more coordinated response through the body. So that's kind of the purpose of that corsum callosum, but it is a solid barrier. And so in between those two halves, we've got what's called the longitudinal fissure. So that longitudinal fissure is that big division, and where they are joined is in the corpus callosum. So that's the first place where we really are structurally held together, if we're looking from the top down, is that corpus callosum. In the center, kind of in the middle there, we've got this other little kind of projection coming down that's called the fornix. And then in the center here is what's called the thalamus. Below the thalamus, this kind of triangular shaped section is the hypothalamus. And then coming off the bottom of the hypothalamus is the pituitary gland. So that pituitary gland, again, if you remember the inside of our skull, we had the sella turcica right there. The pituitary gland sits right in that sella turcica. So that's kind of how this is fitting into the skull. We'll talk about what those things do in a bit. So the diencephalon is essentially everything from the corpus callosum until we get to the midbrain. I think some references might include the midbrain in there, but I think usually the midbrain counts as part of the brain stem. So here's the midbrain that I'm outlining with my cursor right here. And then we go into the pons, and then we go into the medulla, and then we're into the brain, the spinal cord itself. Now the darkest regions in here are basically kind of open space, what are called the ventricles. And so we're going to have cerebrospinal fluid in all of these darkest areas. Now the thalamus is pictured here. Think about the thalamus kind of being, we're going to have a right thalamus and we're going to have a left thalamus, and they're going to kind of touch in the middle. But top and bottom, we're going to have cerebrospinal fluid kind of flowing all around that area to kind of lubricate things. So those are part of our ventricles, which are internal spaces in the brain for that cerebrospinal fluid to go. So that center little dot that's called the interthalamic adhesion, that's essentially like where my knuckles are touching right here. I've got my left thalamus, I've got my right thalamus, they're touching in the middle, but all around there, wherever they're not touching, I'm going to have cerebrospinal fluid going around there. We also have cerebrospinal fluid that's going to lubricate the and come down and lubricate around the cerebellum, as well as all the way down into the spinal cord. So we've got cerebrospinal fluid all the way down. If you've ever heard about people getting a spinal tap, they're drawing out cerebrospinal fluid from the spinal cord to see if there's organisms in there typically. If you've got viral meningitis or bacterial meningitis or something like that. So that fluid does flow all the way through all of the CNS. When we look at the cerebellum that's cut in half, in the center, they call this the arbor vitae, or the tree of life. So you've got this kind of central trunk, and then you've got all these little branches, and then off each branch, you've got kind of these little twigs. So it kind of has that tree-like appearance with that. And that's basically, those are just pathways that carry lots of axons to help us make sense of stuff. At the back of the diencephalon, we've got what's called the pineal gland and the tectal plate. These are some different neurons that help with some motor responses and reflexive responses. The pineal gland is notable because that's where we produce and release melatonin. So if you're familiar with melatonin, a lot of people take melatonin to help with sleep. It is released in response to daylight patterns. And so that's one reason why they tell people not to use like backlit screens at night, because it basically fools your brain into thinking it's still daytime and you won't release melatonin and get sleepy. So it's usually recommended to quit using electronic devices before you're trying to go to bed for that reason. But that's where melatonin's coming from. All right, let's start talking about functions of some of those things. And we'll look at, I know we'll get to a picture where we can see the insula, because I haven't forgotten about the insula. You don't need to worry about any of this developmental stuff on this test. All right, So we've got gray matter, we've got white matter. Most people have heard those terms. If you've watched any kind of detective show or investigative forensic type show, oh yeah, we got gray matter over here. Oh yeah, we got white matter over here. Gray matter is basically parts of the brain that don't have a lot of fat. White matter is parts of the brain that have more fat. So when we have a lot of myelinated axons, that's white matter. Where we have a lot of cell bodies that aren't myelinated, that's gray matter. So gray matter is mostly all the neurons and all the dendrites. And then if we have axons that are not myelinated, they'll also appear grayish on specimens. The white matter is going to be essentially just axons. There's no cell bodies. So there's nothing that's interpreting, generating responses. They're just passing the message on when we have white matter. All right, so when we look at this image, this top one, we've got this coronal cut. So basically I've cut through the brain this way. You can kind of see in the picture here where it is, kind of right through the frontal lobe, kind of coming down is where that image is sliced from. We've got gray matter, and it looks more like beige matter, all the way around the perimeter. That's where all the neurons are. For the frontal lobe, for parietal lobe, whichever part of the brain, whichever slice you're looking at, however far anterior to posterior you're looking at. In this case, it would all be frontal lobe neurons, essentially. In between here, you can see how we've got these cerebral hemispheres dropping down, and then we hit the corpus callosum. So that corpus callosum is a section of axons that are going to carry messages from one side of the brain to the other side of the brain. They're not going out to the rest of the body. They're just going right to left. So it's just all axons right in there. We've also got what are called the internal capsule. Internal capsule are these more diagonal sections. These are going to be the areas of the brain where axons are going to be leaving, going out the brain stem and going down the spinal cord, or if it's sensory information, it's going to be coming up and then going either through that right internal capsule or the left internal capsule. So the internal capsules are the primary highways for messages from the cerebral hemispheres to go in or out. The corpus callosum is that primary passageway just for messages to go right and left within the brain. So those are really critical areas. Now, besides the gray matter that's all around the perimeter, we have some sections of gray matter that are more centrally located. These top two are really, that's kind of where our thalamus is going to be. We're a little bit too far anterior in this particular slice to see the thalamus. So it's what we call cerebral nuclei. These are clusters of cell bodies that aren't really a part of a given lobe. They're more just a part of the brain. But we do still have a right and a left on these, and they control different functions. We'll kind of touch on those a little bit. Right here on the side, these are the neurons that make up the insula. So this little crevice over here, that's that lateral fissure between the temporal lobe and the parietal and or frontal lobe is where that gap is. So all these neurons that are in here, all this gray matter in here, those are the insula. That's that internal lobe of the brain. All right. You'll see in sections of the cerebellum, as well as into the medulla and then into the spinal cord, you do still have sections that are gray and sections that are white. So we do have parts of each of these components of the brain and central nervous system that have neurons themselves, the cell bodies, and then we have other parts that have just myelinated axons. So again, if it's white matter, that's myelinated axons. If it's gray matter, that's either unmyelinated axons or it is cell bodies to control things. All right. So our brain is super important. We can't afford to let it get injured. It's controlling everything else in the body. So if it doesn't work, we don't work. So we definitely don't want it to get injured. So we've got that nice skull to protect it. Pretty strong, but it does get damaged sometimes. We've got the meninges, and so those are basically connective tissue layers that are surrounding the perimeter of the actual neurons and the spinal cord and things like that. We've got the fluid, the CSF, and then we've got those surrounding those capillaries that supply the neurons with blood. We've got those astrocytes that are creating that blood-brain barrier through their little perivascular feet to limit things getting out of the capillaries and getting into the brain itself. So we've got a number of different things set up to kind of keep the brain healthy and keep it from getting damaged, either from internal chemicals, from external forces, or whatever the case might be. All right, so our meninges. We've got 3 layers of meninges: the pia mater, the arachnoid mater, and the dura mater. The pia mater, I wish we still had our samples because we had some that you could really see these different things. The pia mater is like shrink wrap on the brain. If you've ever seen like a piece of meat or fish that's vacuum packed, where it's just the saran wrap type stuff is just suction down and really snug, directly adhering to the product that's inside the packaging, that's how the pia is to the brain. It's a very thin film, like saran wrap, like thick, clear essentially, but it's that first deepest layer that's basically keeping all the CSF kind of just helping hold everything there and kind of keeping the brain, the outer surface of the cortex of the brain, very protected. The arachnoid, and most people know that term related to spiders, it does kind of look spider-like. It's the least dense of the meningeal layers, has a lot of fluid in there, has a lot of blood vessels in there as well. So it is more of a space than a structure almost. And then the dura mater, dura means hard or tough. It is very tough. The dura mater on these things was like a thin piece of leather on the specimens that we had. You could take it with your hands and you could not tear it really with your fingers, I don't think. You'd have to be pretty strong. The pia mater was, yeah, you could tear that pretty easily and pick it right apart. But the dura mater, it's a lot of collagen, it's a lot of fibrous connective tissue, it's very tough. And when I talked about the spinal tap earlier, when a physician is doing a spinal tap or a spinal injection, when they're putting that needle in, they'll feel it when it penetrates through that dura. It's a distinct like pop that they'll feel, that resistance, and then you punch through it. It's pretty easy, I think, for them to know when they've gotten into the space that they're after. So it is a tough layer, and it's a double layer. We've got kind of similar to like all of our visceral layers. We've got an inner layer, and then we've got an outer layer. The inner layer is clinging to the actual arachnoid mater, essentially. The outer layer is basically between the skull or between the inside of the spinal column itself. So we have that double layer to the dura. The arachnoid and the pia just have a single layer to them. All right, so here's a nice little picture of it, kind of looking at the top of the skull, kind of how it's organized. We've got our gray matter on the cortex here. The little purplish line that kind of follows down into all these little grooves, that's the pia. So it doesn't just span the gap where there's a sulcus, it basically follows the sulcus all the way down in. So it is very much shrink wrapped into all those little crevices on the brain surface itself. The arachnoid, we've got all these little vessels located in there, and there is a lot of cerebrospinal fluid that is in there as well. So that's that middle section. And then we've got that double layer of the dura. So especially when you look up here at the top, you can see that double layer because they're kind of back to back to each other. When we get into the central portion, we have the space in there. And so where all that kind of bluish area is, that's going to have a bunch of cerebrospinal fluid. It's also going to have a vein that runs through that area that's going to help pick up some of the peripheral blood that's also going to be picked back up through there. So it has a mixture of CSF as well as veins in there. We've got these, what are called arachnoid villi, and that's basically where the CSF is going to be able to get recycled. So the ependymal cells are going to produce the CSF. They're in certain locations in the brain that we'll see in another image. That CSF flows all through the spaces around that, the brain and the spinal cord, and then it's going to exit through these arachnoid villi, where it's then going to get reabsorbed back into the venous circulation. And we make CSF from blood, and it goes back to being part of the blood as well. If you look in the center here, we don't have a double layer of pia from the periosteal layer as well as the layer on the brain, but we do have basically two layers that are inside. And this particular double layered section that's dropping down into that longitudinal fissure is called the falx cerebri. We'll have another kind of double layer membrane like that going between the two halves of the cerebellum called the falx cerebelli. So that term falx, either cerebri, meaning between the two cerebral hemispheres, or cerebelli, between the two cerebellar hemispheres, that's just a double layer of dura mater between those two, between the right and the left half of those parts. All right, so I just talked about those. The middle term, this tentorium cerebelli, is basically a sheet of dura mater between the cerebellum and the cerebrum. So I'm holding this upside down because otherwise it'll all fall apart. You're looking at the back, so this is the cerebellum. The the tentorium cerebelli would be basically going in between the underside of the cerebral hemispheres and the top side of the cerebellar hemispheres. The falx cerebri or falx cerebelli is going to be going in between, and these are separated, so it will dip in between there a little bit. And then the falx cerebri is going to be in this space right here, that longitudinal fissure. And again, this is upside down. If I held it like a shot put, then you can see it. Otherwise, it'll all fall apart. All right, the diaphragm ocelli is between the pituitary and the hypothalamus, so that's more anterior. But it's essentially similar to the tentorium cerebelli. There's just not a lot of there's not a lot to it. It's pretty small. All right. Finish going through these septa and then see where we're doing time-wise. So this term sinus, don't just think that it refers to here. It's not just that, it's a general term. It just means basically a channel or a tunnel or an open area within the body. So we've got around the brain, we've got the superior sagittal sinus, inferior sagittal sinus, transverse sinus, and occipital sinus. And so those are pretty much telling you where they are. Sagittal is this. Superior sagittal is going to be going right along the top. The inferior sagittal basically goes right above the corpus callosum. So it's going to basically follow right along in here. Superior sagittal, whoops, can't see what I'm pointing at. Superior sagittal sinus is going to be filling this space right there. Inferior sagittal sinus is going to be just above the corpus callosum, but it's essentially a blood vessel and a place for cerebrospinal fluid to be in there. The transverse sinus is going to be going up into this space. Here's the cerebellum. So it's going to be filling this space, essentially where the tentorium cerebelli is. That's where the transverse sinus is going to be. And then the occipital sinus is going to be coming along, again, on the back side, that posterior side. Let's look at what those look like. And these are all just set up to be areas where veins can recollect blood that has been perfusing through the periphery of the brain itself. So we've got our cutaway view here. We've got our superior sagittal sinus where this big blue vein is on the top. We've got our transverse sinus that's kind of below, coming in beneath. Again, our cerebellum is going to be kind of in this space right here. Occipital sinus is going down below, so it's dropping down towards the brainstem. And then the inferior sagittal sinus is going to be here. Again, this part right in here is where the corpus callosum is going to attach the right and the left cerebral hemispheres to each other. So these membranes that are part of the dura mater primarily help create basically sections within the brain. So the cerebrospinal fluid will only kind of circulate inside its own little section, and then it can be passed through other areas, but kind of it creates little compartments. So that the right cerebellum is separate from the left cerebellum, the right cerebral hemisphere is separate from the left cerebral hemispheres. Both cerebral hemispheres are separate from the cerebellum. So these are just, again, connective tissue barriers within the brain. But the purpose, in addition to compartmentalizing different parts of the brain, is to be places where veins can exist and be protected and housed. You can see the pituitary in here, and the diaphragm sella is that more anterior kind of barrier that's going to run horizontally. All right, let's talk about the ventricles, and then we'll probably, maybe the CSF. I don't think we'll get to the brain lobes today. We'll have to hold off on that for next time. All right, so ventricles, those are just spaces where that cerebrospinal fluid is going to flow. And they're hard to visualize in some cases when you're looking at 2D images and looking at models. I kind of like the image on the, well, both of these images kind of give you an idea of where these spaces lie. So any place you see blue, deep internally in the brain, those are open areas where we don't have gray or white matter. It's just going to have cerebrospinal fluid. So we've got cerebrospinal fluid held within the meningeal layers too, that's going to bathe the outside of the brain and spinal cord, but we also want to be able to get that cerebrospinal fluid internally. And so that's the purpose of the ventricles. And so we've got a right and a left. The biggest ones are called the lateral ventricles, and that's kind of all this. It's got this little point that goes kind of back into the occipital lobe, but then it comes anterior as well. When you look at it straight on, it always reminds me of ram's horns, how it kind of curls back like this. Think about I don't know where the Rams are. Where are the Rams located these days? Are they LA or San Diego or wherever they're going to be? I don't know. They've moved so much. But that's what it always reminds me of, those ram horns. In the middle, we have what's called the third ventricle. So we don't have a first and a second ventricle. We don't say that the right is the first and the left is the second. They're both just considered to be lateral ventricles. So you'd have a right lateral ventricle, a left lateral ventricle, but those are essentially the first and second ventricles. The third ventricle connects the right and the left lateral ventricles, so that cerebrospinal fluid can flow back and forth between those areas. And then deeper down below, really more in front of the brainstem, it's actually behind the brainstem. It's between the cerebellum and the brainstem. We have the fourth ventricle, and then again that cerebrospinal fluid is going to flow all the way down the spinal cord to where the spinal cord ends, and the meninges seal off the bottom of the spinal cord, but it circulates around that as well. So that CSF does a lot of important things as far as helping protect the chemical makeup surrounding the brain tissue, but mostly it's just helping be kind of like an airbag. Instead of an airbag, it's like a water bag for the brain, essentially. So the brain doesn't just immediately slam into the skull every time we nod our head, whether it's just a good song or we're saying yes. So it just gives us that buoyancy. And it has its limits. Obviously, people get concussions and brain injuries, so it's not perfect, but it does a pretty good job. The CSF is created in what's called the choroid plexus. And so these are ventricular spaces that you're seeing in this cutaway. And the choroid plexus is in those ventricular spaces. So we make the CSF in that area, and then it can circulate throughout the rest of the central nervous system. Again, these are ependymal cells that are making that secretion, and those ependymal cells are basically a part of that choroid plexus. So we only have a few different locations in the brain where we have them, but essentially where we have those ventricular spaces. All right, if you look at this top image where we were looking at that coronal cut, on either side up top, those are our lateral ventricles, our right and our left, as are the ones over here. So if you remember those ram's horns, this space right here where I've got my cursor is the same ventricle as this space way over here, because it's starts, they're bigger centrally, then they kind of loop out and around and come down in that little kind of hook shape to come into the temporal lobe as well. So those spaces, it's the same space. It's one continuous open space, even though that slice doesn't really show it that way. All right, so you don't need to memorize which direction the fluid flows from. But if you look at the top, this is where we've got our corpus callosum. We've got a thin little membrane in between the right and left halves that's called the septum pellucidum, just below the corpus callosum. That septum pellucidum is a very thin membrane that separates the biggest part of the lateral ventricles. So if we go back a slide, the septum pellucidum is this little thin line that's right in the center of that open space. You can't really see it on here. It's too high up on this picture. All right, we talked about those already. All right, I think we'll quit here. It's 9:30. I think that's when I'm supposed to end. And we'll pick up there and we'll kind of go through the lobes the next time, I guess. We'll start with that. So let's take about 10 minutes. About 9:40, we'll come back. I may just go through the lobes of the brain really quickly then, and then turn you guys loose to just work on some of the handouts, because I think there are a lot of different parts of the brain to try and get straight before the quiz next Tuesday. So I think I'll probably let you guys try and work together during most of the labs this week to try and just help each other learn those structures from the images a little bit better. So take about 10 minutes, about 9, we'll say 9:42, we'll come back and get going on lab.
Hey, Craig, I have a question.
Yep.
If we go back to slide 7, what's the difference between a tract and a nerve?
So when you're looking at the optic in particular, so the optic nerve, the optic nerve's kind of weird. Once we get to this optic chiasm, essentially the nerve itself is getting split. So the information that the retina is picking up from the one eyeball comes in here, and part of that image stays on that side of the brain. Part of the image that this eye picks up crosses over.
Oh, okay.
So when we get past the chiasm, they change the name to tract, because it's not just the information being carried from this eye. Each tract has information from both eyes in it, whereas each nerve, optic nerve, is just for the right eye or just for the left eye.
Okay. Yeah.
But the image, when we think about what our eyes are picking up, if we split it vertically, we have the nasal field of view and the peripheral field of view. Those two halves are carried on opposite nerves, on opposite tracks.
Okay.
And that information goes back to the occipital lobe for us to make sense of what we're seeing.
Okay, makes sense. Thanks.
Yep.
So I got a question.
Sure.
You were talking about that ice pack causing that numbness,
Uh-huh.
damaging that nerve. So I've noticed over the past several months that I've got a little bit of tingling in my left leg, my like third through fifth digits. Not serious. It's like sometimes I can't even tell it's there, and then sometimes it's just really, really light. And that leg has taken a beating. I've had it scoped twice. I had varicose vein repair done on it, but I also wear knee pads at work. I wear very light. The pressure is not light, but I've wondered if wearing those knee pads all day every day isn't putting some pressure on there. But I'm just trying to figure out the source of that numbness, because I don't have any diabetic issues. I don't have any. I mean, everything else is healthy. It's just when you said that, it really kind of tripped me.
Yeah, I had a nursing student when I used to teach anatomy for them a couple of years ago, and she was complaining about the same thing. And I was asking her about, her work and her job and stuff. And she's like, "Oh yeah, well I started wearing compression hose at work because I'm on my feet all the time." And I was like, "Yeah, you're probably that compression hose because of where that, especially that perineal nerve is. It's essentially there's the head of the fibula. If you're getting knee-high compression socks, that thick band is essentially pushing down right on that nerve and pinning it against your underlying bone, essentially. So it is an area where you're susceptible. So if you have knee pads on and they have elastic that goes around the knees to hold them in place, then that elasticity could be enough that you might be getting some compression on that fibular perineal nerve. That could be
no, a double whammy. I am wearing compression stockings too.
Yeah. So, I mean, you could potentially think about trying to maybe roll down the compression sock just a little bit so you get off of the fibular head. The problem with that is it kind of magnifies the pressure where you've doubled it over itself.
Yeah, exactly.
But it's right around that bone is where you're most susceptible to compressing it. If you've got muscle underneath it, then the elasticity can kind of push the nerve into the muscle and the muscle gives. But when you can push the nerve into the bone doesn't give. It's just going to mash on the nerve.
So if I make a change, it's going to take months before.
It could be. I mean, it could be that you might only be causing some ischemia and some nerve compression while you actually have them on.
Oh, you'd have to have nerve damage for it to take a long time.
Right. But it could be that if you're wearing them long enough hours of the day, all the time, that maybe it has caused significant enough damage downstream.
Well, it's 6 days a week, 10 to 12 hours a day.
Yeah. So I mean, it could be that you've caused some permanent damage that might take a little while to wake up, but I would think it also might be that if you relieve that pressure, that it might instantaneously fix it potentially. If you're only having symptoms when you are wearing the compression socks, then for sure I would say try getting that just down a little bit lower. But if you have symptoms all the time, even when you're home and you don't have anything on your legs, then you might have a little bit more permanent or semi-permanent damage that might need some time to recover.
Okay, that all makes sense.
Yeah. All right. So 9:45. I think I'm just going to let you guys work on the handouts, the labeling handouts, because I think that's probably the best use of time. We'll have our quiz starting at 10:20. So it gives you guys about 35 minutes in your small groups to work on that. So I would go ahead and make sure at least one person in your group has that pulled up, the labeling handouts for all this week's lab activities. Should have all these brain structures that we've been going through. If we haven't covered all the material yet, then I wouldn't necessarily work on those. There's a whole bunch of them. I think there's like 15, 16 different images, different angles and things like that. So just work on the ones that we've covered so far, and then we'll finish covering the rest of this chapter on Thursday. And then Thursday's lab session, you can kind of work on the ones that we'll cover a little bit more in depth on that one. But you should have a pretty good start to get a lot of these kind of big picture structures, as well as some of the segments and sections and fissures and sulcuses and all that kind of stuff. So I'm going to go ahead and get your breakout rooms created. I can remember where I go. There we go. We're missing one person, so probably one group will have 2 people, but all the other groups will have 3. All right. So I'm gonna give you guys a few minutes, and then I'll start popping in room by room. right, so we're getting everybody back, looks like. I think once 10:20 hits, which it just did on my computer, you should be able to start your quiz. So just keep your camera on for that. And then once you're done with the quiz, you're welcome to check out. And then next time on Thursday, we'll finish going through all the brain stuff, spinal cord, talk about cranial nerves a little bit, and kind of finish up this chapter. And then I'll review what next week's quiz more specifically is on, but it is basically on all this brain stuff. But I'll let you know how much of the spinal cord related things you need to know. And if you need to know any, I don't think there's any cranial nerve stuff on the quiz. I think it's more just structural stuff. So yeah, like I say, go ahead and get going on your quiz if you want. And let me know if you have issues or if you have questions that popped up during the quiz, then just put them in the chat and I'll get with you on that.